Dear viewers, this video is part 5 in the series on cancer, that is Neoplasia, in the Medicine for Common Man series. So, coming back to the subject, even cells that are not actively growing must shunt some metabolic intermediates away from oxidative phosphorylation to synthesize macromolecules that are needed for cellular maintenance. By contrast, in actively growing cells, only a small fraction of the cellular glucose is shunted through the oxidative phosphorylation pathway, such that, on average, each molecule of glucose that is metabolized produces approximately 4 molecules of ATP instead of the 2 molecules that would be produced by pure glycolysis. Presumably, this balance in glucose utilization, heavily biased toward aerobic fermentation with a bit of oxidative phosphorylation, hits a metabolic sweet spot that is optimal for growth. It follows that growing cells do rely on mitochondrial metabolism. However, the main metabolic function of mitochondria in growing cells is not to generate ATP, but rather to carry out reactions that generate intermediates that can be diverted for use as precursors in the synthesis of cellular building blocks. For example, lipid biosynthesis requires acetyl coenzyme A, and acetyl coenzyme A is largely synthesized in growing cells from intermediates such as citrate that are generated in mitochondria. So, how is this reprogramming of metabolism triggered in growing normal and malignant cells? As might be guessed, metabolic reprogramming is produced by signaling cascades downstream of growth factors receptors, the very same pathways that are deregulated by mutations in oncogenes and tumor suppressor genes in cancers. Thus, whereas in rapidly growing normal cells, aerobic glycolysis ceases when the tissue is no longer growing. In cancer cells, this reprogramming persists due to the action of oncogenes and the loss of tumor suppressor gene function. So, dear viewers, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions, available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Coming back to the subject, autophagy. Autophagy is a state of severe nutrient deficiency in which cells not only arrest their growth but also cannibalize their own organelles, proteins, and membranes as carbon sources for energy production. If this adaptation fails, the cells die of starvation. Tumor cells often seem to be able to grow under marginal environmental conditions without triggering autophagy, suggesting that the pathways that induce autophagy are disabled. In keeping with this, several genes that promote autophagy are tumor suppressors, meaning that the loss of autophagy enhances tumor growth. Under conditions of severe nutrient deprivation, tumor cells may use autophagy to become dormant, a state of metabolic hibernation that allows cells to survive hard times for long periods. Such cells are believed to be resistant to therapies that kill actively dividing cells and could therefore be responsible for therapeutic failures. Thus, autophagy may be a tumor's friend or foe depending on how the signaling pathways that regulate it are wired in a given tumor. Altered cellular metabolism overview. Warburg metabolism is a form of pro-growth metabolism favoring glycolysis over oxidative phosphorylation. It is induced in normal cells by exposure to growth factors and becomes fixed in cancer cells due to the action of certain driver mutations. Mainly, oncoproteins, RAS, MYC, mutated growth factor receptors, induce or contribute to Warburg metabolism, and many tumor suppressors, PTEN, NF1, and P53, opposite. Stress may induce cells to consume their components in a process called autophagy. Cancer cells may accumulate mutations to avoid autophagy or may corrupt the process to provide nutrients for continued growth and survival. Some oncoproteins, such as mutated IDH, act by causing the formation of high levels of oncometabolites that alter the epigenome, thereby leading to changes in gene expression that are oncogenic. Note: isocitrate dehydrogenase 1 and 2, that is IDH 1 and 2, are key metabolic enzymes converting isocitrate to alpha ketoglutarate, that is AKG. IDH 1 and 2 mutations have been identified in multiple tumor types, including gliomas, a type of brain tumor, and myeloid malignancies such as acute myeloid leukemia, AML. Evasion of cell death. Tumor cells frequently contain mutations in genes that result in resistance to apoptotic cell death. Apoptosis, or regulated cell death, refers to an orderly dismantling of cells into component pieces, which are then efficiently consumed by macrophages without stimulating inflammation. There are two major mechanisms by which apoptosis is avoided by cancer cells. Loss of TP53 function. Note: the TP53 gene provides instructions for making a protein called tumor protein, that is P53, or that is protein 53. This protein acts as a tumor suppressor, which means that it regulates cell division by keeping cells from growing and dividing, that is proliferating, too fast or in an uncontrolled way. TP53 is commonly mutated in cancers. Overexpression of anti-apoptotic members of the BCL2 family. Overexpression of BCL2 is a common event leading to the protection of tumor cells from apoptosis. One of the best understood examples is found in follicular lymphoma. A B cell tumor, it is a type of lymphocyte carrying a characteristic 1418 Q32 Q21 translocation that fuses the BCL2 gene to the transcriptionally active immunoglobulin heavy chain, that is a type of protein involved in immunity, heavy chain gene. The resulting overabundance of BCL2 protects the transformed lymphocytes from apoptosis. Because BCL2 overexpressing follicular lymphomas arise in large part through reduced cell death rather than explosive cell proliferation, they tend to be indolent, that is slow growing. Evasion of cell death. Overview. Apoptosis can result in the activation of a proteolytic cascade of caspases that destroys the cell. Lesions that incapacitate the intrinsic, that is the mitochondrial pathway, appear to be most common in cancers. In greater than 85% of follicular B cell lymphomas, the anti-apoptotic gene BCL2 is overexpressed due to a 1418 chromosome translocation. Limitless replicative potential, the stem cell-like properties of cancer cells. All cancers contain cells that are immortal and have limitless replicative potential. Some cell lines established from cancers have now been proliferating ceaselessly in laboratories for more than 60 years. How can it be that cancer cells have seemingly discovered the proverbial fountain of eternal youth? The answers are not completely known, but three interrelated factors appear to be critical to the immortality of cancer cells. 1. Evasion of senescence. 2. Evasion of mitotic crisis. And 3. The capacity for self renewal. 1. Evasion of senescence. Most normal human cells have the capacity to divide 60 to 70 times. After this, the cells become senescent, permanently leaving the cell cycle and never dividing again. The mechanisms that produce senescence are not well understood, but the senescent state is associated with upregulation of P53, and by maintaining RB, that is retinoblastoma protein, in a hypophosphorylated state, which favors cell cycle arrest. See figure 1.5, the cell cycle shown earlier. The RB dependent G1S cell cycle checkpoint is disrupted in virtually all cancers by a wide variety of acquired genetic and epigenetic aberrations that may allow cells to bypass senescence. 2. Evasion of mitotic crisis. While cells that are resistant to senescence have increased replicative capacity, they are not immortal. Instead, they eventually enter a phase referred to as mitotic crisis and die. Mitotic crisis has been ascribed to progressive shortening of telomeres, special DNA sequences at the ends of chromosomes. Most somatic cells do not express telomerase, the enzyme that is responsible for the maintenance of telomeres, and with each cell division, their telomeres shorten. When the telomeric DNA is completely eroded, the exposed chromosome ends are sensed as double-stranded DNA breaks. If the affected cells have functional P53, the cell arrests its growth and may undergo apoptosis. But if P53 is dysfunctional, the end joining pathway is activated and may join the naked ends of two chromosomes. The resulting dicentric chromosomes are broken during attempted mitotic segregation during anaphase, producing new double-stranded DNA breaks. The snowballing genomic damage eventually produces mitotic catastrophe and cell death. Figure 1.7 shows a dicentric chromosome. It has two centromeres with its own chromosomal segments. It is formed by the fusion of two chromosomal ends. Ultimately, the chromosome, which is dicentric, leads to chromosomal instability. See figure 1.7 dicentric chromosome. Note, a dicentric chromosome is an abnormal chromosome with two centromeres. It is formed through the fusion of two chromosome segments, each with a centromere. Telomerase is expressed at very low levels in most somatic cells, and thus proliferating cells that escape from senescence are likely to expire in this fashion. However, cells in crisis that reactivate telomerase can restore their telomeres and survive. Such cells may have suffered damage to oncogenes and tumor suppressor genes and are at high risk for malignant transformation. Alternatively, cancers may arise from stem cells which are long-lived in part because they express telomerase. Whatever the mechanism, telomere maintenance is seen in virtually all types of cancers. In 85 to 95% of tumors, it is due to expression of telomerase. Self-renewal. Unlike most cells, tissue stem cells and germ cells express telomerase, making them resistant to mitotic crisis and also somehow avoid the genetic and epigenetic alterations that trigger senescence. In addition, long-lived stem cells possess another critical property, is capacity for self-renewal. In simple terms, self-renewal means that each time a cell divides, that is a stem cell divides, at least one of the two daughter cells remains a stem cell. In a symmetric division, both daughter cells remain stem cells. Such divisions may occur during embryogenesis, when stem cell pools are expanding, or during times of stress. In an asymmetric division, only one daughter cell remains a stem cell. In such circumstances, the non-stem cell daughter proceeds. The non-stem cell daughter proceeds along some differentiation pathway, losing stemness but gaining one or more functions in the process. Cells in transit to a differentiated state are often highly proliferative, but they eventually differentiate, stop dividing, and may eventually become senescent or undergo apoptosis. The continued growth and maintenance of many tissues that contain short-lived cells, such as the formed elements of the bone marrow and blood, and the epithelial cells of the gastrointestinal tract and skin, depend on a resident population of tissue stem cells that are capable of self-renewal. Because cancers are immortal and have limitless proliferative capacity, they too must contain cells that self-renew, the so-called cancer stem cells. Cells resembling stem cells must exist in all cancers. Another open question is whether cancer stem cells arise from the transformation of tissue stem cells or from the conversion of conventional somatic cells to transformed cells with the acquired property of stemness. The answer seems to be that both scenarios occur in different types of tumors, as exemplified by chronic myeloid leukemia and acute promyelocytic leukemia. Cancer cells within a tumor may be able to de-differentiate to a stem cell-like state. Indeed, there is evidence that cancers can repopulate their stem cell pools from non-stem cell populations, further complicating efforts to precisely define and selectively target cancer stem cells. Despite these uncertainties, the concept of cancer stem cells has important implications for cancer therapy. Most notably, if cancer stem cells are essential for tumor persistence, it follows that these cells must be eliminated to eradicate the tumor. It is hypothesized that, like normal stem cells, cancer stem cells have a high intrinsic resistance to conventional therapies due to a low rate of cell division. Thus, the limited success of current therapies may in part be explained by their failure to kill the malignant stem cells that lie at the root of cancer. Limitless replicative potential overview. At least some cells in all cancers must be stem cell-like. These cells are sometimes referred to as cancer stem cells. This may arise through transformation of a normal stem cell or through acquired genetic lesions that impart a stem cell-like state on a more mature cell. Cancer cells acquire lesions that inactivate senescent signals and reactivate telomerase, which act together to convey limitless replicative potential. Angiogenesis. Even if a solid tumor possesses all the genetic aberrations that are required for malignant transformation, it cannot enlarge beyond 1 to 2 mm in diameter unless it has the capacity to induce angiogenesis. Like normal tissues, the health of tumors requires delivery of oxygen and nutrients and removal of waste products. Presumably, the 1 to 2 millimeter size is the limit for the maximal distance across which oxygen, nutrients, and wastes can diffuse from existing blood vessels. Growing cancers stimulate neoangiogenesis, during which new vessels sprout from capillaries. Neovascularization, that is, new vessel formation, supplies needed nutrients and oxygen. Proliferating endothelial cells also stimulate the growth of adjacent tumor cells by secreting growth factors such as insulin-like growth factors and PDGF. Note, insulin-like growth factor 1, that is IGF-1, resides on the long arm of chromosome 12, also called somatomedin C, is a hormone structurally similar to insulin, plays an important role in childhood growth, and has another anabolic effects in adults. IGF is a protein encoded by the IGF-1 gene. Produced primarily by the liver, production is stimulated by growth hormone. IGF-1 is produced throughout life. The highest rates of IGF-1 production occur during the pubertal growth spurt. The lowest levels occur in infancy and old age. Platelet-derived growth factor, PDGF, regulates cell growth and division. PDGF plays a significant role in new blood vessel formation, proliferation of mesenchymal cells such as fibroblasts producing fibrous tissue, osteoblasts producing bone, vascular smooth muscle cells, and mesenchymal stem cells. Chemotaxis, the directed migration of mesenchymal cells. Though PDGF is synthesized, stored in the alpha granules of platelets, and released by platelets upon activation, it is also produced by other cells, including smooth muscle cells, activated macrophages, and endothelial cells. While the resulting tumor vasculature is effective at delivering nutrients and removing waste, it is not entirely normal. The vessels are leaky and dilated and have a haphazard pattern of connection. By permitting tumor cells ready access to these abnormal vessels, angiogenesis also contributes to metastasis. Angiogenesis is thus an essential facet of malignancy. How do growing tumors develop a blood supply? Angiogenesis is controlled by a balance between angiogenesis, promoters, and inhibitors. In angiogenic tumors, this balance is skewed in favor of promoters. So, dear viewers, this video is part 5 in the series on cancer, that is neoplasia, in the Medicine for Common Man series. Thank you for watching the video.